Influenza is a single-stranded spherical RNA virus that is a part of the Orthomyxoviridae family. The influenza virus can be categorized into three types, A, B, and C, which were named in the order they were discovered. Type C is generally rare as it infrequently causes human infection and doesn't contribute much to human infection studies. Type B has a milder infection course and slowly spreads but can still cause epidemics. Lastly, type A is the most dangerous and common among the types of influenza due to its high mortality and morbidity. Additionally, the virus's protein, hemagglutinin, and viral interface, neuraminidase, make up the outside shell of the virus. The pattern and the shape of hemagglutinin and neuraminidase can further categorize the virus into subtypes. The ones known to infect humans are H1, H2, and H3, and N1, N2, and N3.
Influenza is a very contagious respiratory illness. The influenza virus commonly attacks the epithelial cells that are found in the respiratory lining from your nose to your lungs. The other components of the virus shell are known to be responsible for the infection of epithelial cells. The spike extensions known as hemagglutinin binds to a type of a receptor of respiratory cells that usually binds with sialic acid. The bonds between the virus and the cells introduce the cells to a coat of the virus with a membrane that transports the virus inside the cells. Once inside, the virus undergoes a process that makes the cells produce a new copy of the virus. These new copies use the viral interface neuraminidase to turn off sialic acid receptor. Neuraminidase is also tasked with cutting these new virus copies free from the infected cells so they can find more cells to infect.
The infected cells are identified, the immune system initiates the primary innate immune response, which includes the elimination of microbial invaders by phagocytes and natural killer cells. These phagocytes eliminate these foreign entities and digest them. They release a molecule called interleukin-1. Additionally, the destruction of these cells causes a buildup of mucus, which is then expelled through the cilia of these cells. This results in a high concentration of interleukin-1, which causes the body to induce a fever and the expulsion of mucus through sneezing and coughing. However, the speed of replication and the number of viruses created is too much for the innate immune system. Thus, a specific immune response is required. And these are the lymph nodes containing T and B cells, which are able to produce specific antibodies to effectively identify and neutralize the virus.
When we become infected with the influenza virus, the virus will internalize into our host cells, releasing its viral genome. The viral genome will then use mechanisms to transcribe and translate viral glycoproteins such as hemagglutinin and neuraminidase. These glycoproteins are in the plasma membrane where the viral genome will then travel to perform viral progeny assembly, to bind the glycoproteins to the viral genome while simultaneously releasing the genome into the extracellular space, allowing for the spreading of the virus. On the plasma membrane of the cell, there are sialic acid receptors, which will bind to the hemagglutinin, keeping the virus attached to the surface of the host cell, not allowing the virus to spread. While the virus is bound to the sialic acid receptors, neuraminidase will cleave the sialic acid receptors, releasing the virus from the host cell to begin spreading. Drugs such as zanamivir are neuraminidase inhibitors that will attach to the neuraminidase to disrupt the cleaving of the sialic acid receptors, keeping the virus attached to the host cell, disrupting the spreading of the virus, allowing our adaptive immune system to come in and eliminate the virus.
One important aspect of influenza global surveillance is the detection of influenza strains that are less susceptible to neuraminidase inhibitors. The World Health Organization's global influenza surveillance and response system laboratories around the world typically utilize the neuraminidase inhibitor assay to test for the IC50 value of the specified NAI in question. The IC50 value is defined as the dosage of the drug required to inhibit neuraminidase enzyme activity by 50%. The NAI assay is a fluorometric enzyme assay that measures enzyme activity using a fluorescent product. The light is then measured to determine the activity of the enzyme. In order to analyze this data, the IC50 value that was experimentally determined is compared to reference IC50 values for a specified type or subtype of a virus in order to determine any increased or decreased susceptibility to the NAI drugs. Reduced enzymatic inhibition is typically characterized by changes in amino acids in or near the neuraminidase active site, which does in turn affect NAI binding.